Hi, thank you very much for joining us today. My name is Philippe Trochet, and I'm an application specialist at FUJIFILM VisualSonics. It's a pleasure to introduce to you our high-frequency ultrasounds on photoacoustic modalities for early tumor detection and tumor metabolism studies. For those who may not know VisualSonics, it's a Canadian company based in Toronto, and VisualSonics is part of the medical division of FUJIFILM. The Vivo system is our multimodal imaging platform. It's a fantastic research tool that can be used for many different application areas, for cardiovascular research, development biology, nephrology, neurology. But today I'm going to focus my presentation of what the system can do for oncology. And as you can see here on the circle, the system has many features, allowing to detect tumor at an early stage, using measurement tools for the tumor sizing, having some imaging modes for studying the tumor angiogenesis, detecting the tumor hypoxia, or simply look at the biodistribution of a contrast agent. You can also implant tumor cells on drug delivery. So why to use ultrasound on photoacoustics? These two imaging modalities are compliant with the 3 Rs: reduce, reuse, and recycle. These are two high-resolution imaging techniques. It's in vivo and real time. These techniques are non-ionizing techniques, so it's safe for the animal, and you can do longitudinal studies. And finally, it's very cost efficient. The cost of the consumables is very cheap and low in maintenance demand. How does microultrasound work? So in fact, it's very simple. As you can see here on the video, we have a mouse having a tumor on its hind limb, and we have this line array transducer, which consists in 256 nanocrystals, and each element emits a wavesound and receives a wavesound back from the tissue. So it's real time. And if you attach the transducer to its 3D motor, you can scan the tumor in a stepwise manner, and the step size can be as low as 80 micrometers. So the system takes multiple slices, and at the end of the 3D scan, it stitched together all the images. And by loading the CineLoop into the 3D software, you get right away the 3D volume construction. You may wonder what's the difference between a conventional clinical ultrasound system that you find in hospitals and the Vivo system. So as you can see here, a clinical system works in a frequency range from 3 to 15 megahertz, and the resolution is about 200 to 300 micrometers on the image. While the Vivo system works at a frequency range which is much higher, up to 70 megahertz, and typically at 50 megahertz, the resolution is 30 micrometers. So high frequency means high resolutions. However, at high frequency, the wavesounds penetrates a bit less into the tissues, but it fits perfectly when imaging small animals such as mice, rats, rabbits, or many other animal species. We have different transducers working at different central frequencies, and depending on the application area or the animal model, you can select the transducer of your choice. At Fujifilm VisualSonics, we have experience scanning mice and rats, but thanks to the researchers, sometimes to time we also have to work on other animal species. And it's really interesting to see what the system can do when looking at octopus or salamander, monkeys, fish, rabbits, and many other animal species. Because the animal model can be small, we like using this imaging platform. So we put the animal on a heated plate, and we place the animal paws on electrodes. And this way we can monitor the physiological parameters such as the animal heart rate and respiration rate. And when inserting a rectal prod, we can also measure the animal body temperature. So here the transducer is attached to its 3D motor, and you can easily tilt and rotate the 3D motor to get access to any part of the mouse anatomy. And we have also some micro manipulators to fine-tune a little bit the animal positioning for the best image field of view. On the left side of this imaging platform, you have the microinjection mount. So if you want to implant tumor cells or do biopsies, you can insert any type of devices. And at the bottom right, here you see our two systems, the Vivo 3100 and the Vivo Laser X. With the Vivo system, you can extract many different parameters to get anatomical, functional, and molecular information. You can detect tumor at a very early stage, and we have measurement packages available, allowing to do very accurate linear measurements, as well as measuring the tumor volume. For functional imaging, we like using our Doppler modes to visualize the tumor vascularization and get the percent vascularity calculated. And to enhance the microvascularization, we like using non-targeted contrast agents where you can do perfusion kinetic studies and quantify some specific contrast parameters. For molecular information, you can detect the endogenous contrast of the blood and quantify the hemoglobin content or the oxygen saturation level for tumor hypoxia. And using exogenous contrast, you can have organic or inorganic contrast agents that are fully compatible with our techniques. And this way, you can use targeted contrast agents to express specific receptors, as an example. We have researchers using the system to implant tumor cells or drug delivery. Here is an example on how you can implant tumor cells into the pancreas. And you can be very accurate in inserting the needle tip into the pancreas. And once you inject the cells, you can visualize the cells diffusing into the pancreatic tissues. And after that, you can start doing a longitudinal study by visualizing and measuring the tumor growth. How can we detect tumors at a very early stage? Each organ in the animal body has its own tissue structure. So here, as an example, if we look at the control animal, the mouse liver has a very smooth tissue structure, very uniform in gray level. And please note the scale. The scale is in millimeters, so even half a millimeter is very big on the image. So you won't miss any abnormalities when looking at a diseased animal. You can identify liver tissue fibrosis or liver lesions, or here, as an example, liver metastases. And this is applicable on all type of organs. Can be the kidney, the pancreas, the prostate, ovaries, and so on. The Vivo product is a true two-dimensional and three-dimensional imaging system. In this paper, researchers show how complementary can be high-frequency ultrasound imaging and optical imaging. Bioluminescence and fluorescence techniques are very sensitive. You can really detect tumor cells in a cluster deep into the tissues. However, the light is highly scattered when diffusing into the animal body, and there's a real lack of resolution. So when combining both techniques and using our high-frequency ultrasound transducers, you can really see where is the tumor into the animal body, how deep is the tumor, if there's one or multiple tumor spots, or eventually metastasis surrounding the primary tumor, and then you can get the tumor volume calculation. Also, some studies have reported how complementary can be high-frequency ultrasound with MRI. And here, it's a good example on showing how good is the correlation of the volume calculations with the two imaging modalities. Some tumor types may seem to be challenging to detect. And here is a good example on how quick and easy can be the colon tumors detection. You simply have to fill the colon with gel, and then you scan the whole colon area in 3D. And this way you can detect the tumors and get the tumor volume calculation. This is a brief overview of the tumor types that the system can detect. but at FUJIFILM VisualSonics, we have some experience looking at lung tumors, liver tumors, pancreatic, colon, prostate, renal, and also brain tumors. And now you may be keen to understand how the system can detect vessels into the tumor tissues, as well as how it can assess the oxygen saturation level into the hemoglobin. Here the mouse has a tumor on its hind limb, and when the tumor grows, the tumor needs to be supplied in blood and nutrient. So there's a vessel growth. For detecting the blood vessels, we use the power Doppler mode, which is sensitive to the blood motion into the vessels. So without the use of any contrast agent, you can detect the vessels networks in the entire 3D tumor volume. The system then does the calculation of the percent vascularity into the tumor volume. So using the power Doppler mode, you can do a quick screening of your mice by detecting the vasculature without the use of any contrast agents. As an example here, you see that at day 1, there's a PV percent of 3.18%, but then 4 days later on, you see that the vessel networks has increased and the PV percent has also increased. Here is a good example of the use of the color Doppler mode in 3D when testing an anti-cancer drug. So you see that after 24 hours, the drug injections, there's a reduced blood flow into the vessels. We now know that power Doppler mode can be used to detect arteries, veins, arterioles, and venules. But what about these tiny capillaries that are formed during the neoangiogenesis? These capillaries are 5 to 10 micrometers in size, and the blood motion is too slow to be detected in power Doppler mode. So to enhance the microvasculature, we like to use a contrast agent which is ultrasound compatible. This contrast agent consists in microbubbles that are 2 to 3 micrometers in size. The microbubbles are filled with perfluorocarbon gas and have a phospholipid shell, so they clear out from the animal body after 30 minutes. The bubbles can be non-targeted and targeted microbubbles. And they are made by Braco for FUJIFILM VisualSonics. They have been fully optimized and are very efficient in the frequency range of our transducers. On these images, you can visualize the microbubble perfusion after a bolus injection. And please note the resolution, which is now the resolution of the microbubble itself. So you can really visualize single microbubbles perfusing into these very tiny capillaries. And the microbubbles are 2 to 3 micrometers in size. We do have a contrast quantification software, the VisualSonics post-processing software. It shows in a color-coded parametric map the microbubbles perfusion, and you can see here in red the best perfused areas and in blue the lower perfused areas. You can extract many different contrast parameters such as the peak enhancements for the relative blood volume or the washing rate for the relative blood velocity. And all these values are reported in the report page and can be exported into an Excel sheet. The targeted microbubbles have a streptavidin coating, so you can simply conjugate any type of bioutilated antibodies. It's really straightforward. You simply had to add a certain concentration of bioutilated antibodies into the microbubble vial. And at FUJIFILM VisualSonics, we have some experience expressing receptors such as the VEGFR2 receptor for tumor angiogenesis studies or the pacemaking receptors for inflammation model. We do have experience as well expressing PCAM or VCAM receptors. Still using the VisualSonics software for contrast quantification, we can do a relative quantification of, in this example, the VEGFR2 receptor. And prior to the injection of the primary antibody expressing the VEGFR2, we inject an isotope control to express the non-specific binding. This was a quick overview showing how the Vivo system in high-frequency ultrasounds can help researchers to extract anatomical functional information as well as some molecular information with the targeted microbubbles. The system can do much more. And now I'm going to introduce to you the photoacoustic imaging modality, allowing to do even more in molecular imaging. FUJIFILM VisualSonics launched on the market the first photoacoustic imaging system commercially available. And now the technique is well established in the core facility lab, especially in oncopharmacology, where you can find optical imaging system as well as PET, SPECT, CT, MRIs, and high-frequency ultrasound imaging systems. One of the main applications in photoacoustics is the detection of the hemoglobin content and the oxygen saturation level in tumors. The photoacoustic phenomenon is a combination of light with ultrasounds. So we have here our high-frequency ultrasound imaging system combined to a laser. And these are the main specifications of the system. Nowadays, the Vivo F2 Laser X works in a frequency range from 1 MHz to 71 MHz. And this way we can scan small and large animal models. The photoacoustic phenomenon is a combination of light converted in wavesounds. So we have here optical fiber that is connected to a tunable laser, and we can select a specific wavelength at which, as an example, the oxyhemoglobin or deoxyhemoglobin absorbs the light. So when absorbing the light, there's a thermoelastic expansion phenomenon that is produced, and this phenomenon is converted into a wavesound. So to summarize, you send lights and you detect wavesounds. And we know at which wavelengths the deoxyhemoglobin absorbs the light and at which the oxyhemoglobin absorbs the most. And when absorbing a lot of light, there will be a high photoacoustic emission phenomenon that is produced. On detecting the deoxy and oxyhemoglobin, the system does the calculation of the oxygen saturation percent level into the hemoglobin. So when this is applied to tumor imaging, using the oxyhemo mode, you can detect the tumor hypoxia. And here, as an example, you can visualize the hypoxic core of the tumor. And this is really important because when testing a therapeutic treatment, it's really important to test the treatment efficacy prior the tumor hypoxia, and also during the hypoxia and before necrosis, when the tumor is more resistant to treatment or also more aggressive. And here we have a co-illustration of the B-mode image, which is the ultrasound image, and the photoacoustic signal. And you can overlay both, and you know where the photoacoustic signal comes from. And the photoacoustic mode is 2D and 3D compatible, so you can measure, calculate the SO2 average percent of the hypoxic core, as well as in the total tumor volume. Oxygen saturation measurements have been validated in comparison with different techniques. And here we have an example when comparing the photoacoustic image with histology hematoxylin eosin. And in the red shows the total hemoglobin content, where the oxyhemo images shows the percent of oxygenation of the hemoglobin. And this matches with the histology slice. This has also been compared with hypoxia detected by immunofluorescence visualization. And here you can see that there's a perfect match between the images. This example shows the benefit of using a bimodal imaging system where we have the high-frequency ultrasound image where we can identify the tumor and measure the tumor volume. So we see that the tumor volume is increasing over time. And this is correlated with the photoacoustic image showing the oxygen saturation map. And here we can detect that the tumor has reached a hypoxic stage, and you can visualize in a very detailed manner the hypoxic area here. So again, this is essential when testing a therapeutic treatment, when we know that when the tumor becomes hypoxic, the tumor is more aggressive and more resistant to treatment. This is another example of the benefit of combining different imaging modes. So on the first image here, you see the microbubbles perfusion map, and you see that there's a region of interest that is not perfused. But here, I can't tell you if this area is a necrotic area or if it's a hypoxic area. Because when the vessels gets damaged, when the microbubbles pass through the vessels, they are destruct because the shell of the microbubbles is fragile. But when you switch to the oxyhemo mode to look at the oxygen saturation level, you see that this area is poorly oxygenated. So then I can suspect that this is a highly hypoxic area. And when you switched again to the total hemoglobin mode, you see that this area is really hypoxic, and there's a lot of blood content. And this simply shows that the blood is stagnating in this area, and this area will turn quickly to necrosis. You can find in the literature and also on our website very relevant papers showing the correlation when comparing oxygen saturation measurements in photoacoustic mode with bold MRI, as well as other papers predicting the response to therapy when measuring oxygen saturation into tumor tissues. Researchers have also reported that photoacoustic imaging can be used to detect brain tumors, and also can differentiate primary brain tumors from secondary brain tumors. So here, as an example, we are looking at melanoma tumors, and we have the control animal on the tumor mouse model. And here is also a good example on how you can use the different imaging mode on the Vivo system to extract different parameters. So as an example, we have this color Doppler mode in 3D showing the main vasculature. And when you compare it to the tumor model, you see that there's some discrepancies in the vasculature network, and this is due to the tumor which is pressing on the vessels. And then you can quantify the hemoglobin content. So there's here much more hemoglobin into the tumor tissues compared to the control animal. They also did oxygen saturation measurements, and you see that the healthy tissues are higher oxygenated than the tumor tissues. And finally, the melanoma tumors produces melanin. So using the Vivo Laser X, we can detect the melanin at a specific wavelength, and doing spectral mixing, we can identify the tumor spot and see how deep is the tumor and what's the tumor volume. So far, I have introduced to you how endogenous contrast can be detected in photoacoustic mode. But there's also exogenous contrast agents that are photoacoustic compatible. Here is the list of contrast agents that are photoacoustic compatible. So you can have organic probes that have very good photoacoustic properties, as well as nanoparticles. So everything that is graphene coated, nanocarbon tubes, gold nanorods, iron oxide nanoparticles show a strong photoacoustic compatibility. Considering organic contrast agents, the ICG is considered as a gold standard in photoacoustic imaging. But recently, there's new dyes that are available on the market, and especially the Angiostamp 800 is really interested because it's targeted contrast agent targeting the alpha beta 3 integrins, a cell surface receptor involved in the cell adhesion and migration. And you can use this Angiostamp as a tumor growth marker. And this has been validated for many different tumor cells. It has been proven that the Angiostamp 800 can target different tumor types, such as ovarian tumors, pancreatic tumors, breast, bone metastases as well, lung tumors, and so on. I hope you enjoyed this presentation and that I have been able to show you that high-frequency ultrasounds and photoacoustic imaging modalities can be useful for your research. Please visit our website, VisualSonics.com. There's a lot of information available, and it will be a great pleasure to stay in touch with you. Thank you for your attention, and we hope to talk to you soon.